In Chapter 8, we looked at the different types, or at least a few different types, of microbial metabolism, both aerobic cellular respiration and fermentation. So it seems odd that Chapter 9 will be called microbial growth, but now we're going to be looking at how that metabolism leads to different types of growth, the rates of growth, and the ways that we can detect that. Microbial growth is very important in medicine. We want to prevent unwanted microbes from growing in or on us. The left side shows a biofilm of Staphylococcus aureus that have developed on a catheter, an indwelling catheter. So a tube inserted into a patient's body. Definitely don't want that. So the way to prevent that would be to use a sterile catheter and also gently and thoroughly clean the opening where that catheter is inserted. But if the patient had Staph aureus as part of their normal microbiota, then that could get introduced. It wouldn't have to be from some sort of poor hygiene. And on the right, we've just got a garbage can that was collecting rainwater, and we've got some sort of green biofilm with unidentified microbes growing on the surface. It's probably a combination of algae and other protists, and probably even some bacteria. We've looked briefly at how bacteria divide. The process is called binary fission because it just splits in two. So binary having two parts and fission to split. All organisms, when they reproduce, whether it's at the cellular level, have to make a copy of their DNA. And in prokaryotes, it is comparatively simple to eukaryotes. The DNA has to be duplicated. The chromosome in prokaryotes is a circular chromosome. Then the cell wall and cell membrane have to start to pinch in, and then we get two individual cells that, in the absence of mutation, will be genetically identical. Since prokaryotes don't do sexual reproduction, it's only asexual by binary fission, the offspring will always be genetically identical to the originating cell. Because their genomes are relatively small and simple, if there are any changes in that nucleotide sequence, whether by mistakes during replication, the presence of some sort of chemical, or just happenstance, then we will have a mutation, some difference, and we may not get two identical cells. Populations is how prokaryotes reproduce, right? When we have one offspring, we change the total number in the population, but we look at growth on an individual basis. Growth in terms of a prokaryotic population is not the growth of an individual cell, but the growth of the population of cells. So if we start with one cell, in one generation we have two cells. Each of those divides, so in 2 generations we have 4 cells, etc. And if we kept doing that, it would not take very long for there to be millions to billions of cells in a 24-hour period. All of those plates and test tubes and auger slants that we see in the lab slides, those were always incubated for 24 hours. And when the initial inoculation happens, whether it's dipping the loop into a broth or streaking it across an agar surface, it's absolutely invisible until incubation happens and the microbes that we put into or onto that media undergo growth and cell division. And by the end of it, we get very macroscopic visible growth. Turbidity in a broth or that visible clumps on the surface of an agar slant or an agar plate. This shows the general shape of a bacterial growth curve. We have the logarithm of living bacterial cells, so 1 unit would mean a 10-fold change, and we have time. There are no actual numbers, no values put on these axes because it varies. It depends on the species that's present. How long does it take to undergo a single cell division? It also depends on the environmental factors. What's the nutrients that are available? What's the temperature that's present? What's the pH? How much oxygen? So if the conditions are ideal, then the length of any one of these stages will be very different than if there is some factor that could affect the growth that is not ideal. But the overall shape of the curve will always look like this. The initial stage is called the lag phase, where the organisms are in or on the media and taking time to sense that environment, sensing the temperature, the pH, the amount of nutrients, the amount of space available for them to divide in. In the log phase, also called the exponential phase, we have that most rapid growth, that on a consistent time basis, every unit of that time, we double the population. As a reference point, E. coli, if we provide those cells with ideal growth conditions, it takes about 20 minutes to complete a population round. So within 20 minutes, the population doubles. So within 40 minutes, we'd have 2 divisions, and we'd go from 1 cell to 2 cells to 4 cells. And every single 20 minutes, we would double that entire population as long as we have the correct nutrients and temperature and all the other environmental factors. When that starts to change, we reach the stationary phase. Even when we start with the most ideal conditions, we aren't going to be able to continue growing at that exponential rate, particularly in a closed system, like a test tube with broth, or a test tube with an agar slant, or a petri dish. We are not providing additional nutrients as the ones that were initially there get used up. We're not removing waste as that accumulates. As the population grows, they are also taking up space, and we're running out of room for more bacteria to grow. So at this point, it's not that everything is dying, but the rate of cell division and the rate of death is about equal, so it's flat. And then in the death or decline phase, we are getting that exponential decrease. There are more cells dying than there are cells dividing. And in our closed systems, eventually there would be all dead cells because we're not removing the waste and we have depleted the nutrients. That's why if we were in a face-to-face class, you would have gotten your unknown test tube, an agar slant with your organism growing in it, and every week you would have practiced your aseptic technique and taken cells from that slant and put it onto 2 new slants. And once your new ones grow, we would get rid of the old one because it's been growing for a couple of weeks. It's running out of space and food and everything else. And you would keep your 2 new slants for a week, and at the end of that week, once again, transfer them to new media. Because we don't want them to get past this stationary phase. We would always want to keep a good proportion of living, happy, growing cells so that they could demonstrate their metabolism in all of the different tests we do. If we were to count the number of cells present, we could depict it 2 different ways. I am not going to make you repeat this, but this is showing if we just did a arithmetic scale, then we get it shooting up because every single time unit we are doubling the amount of organisms present. If we do a semi-log scale, meaning we keep the time linear, but the number of cells logarithmic, then it appears to be more of a linear graph. And that just makes it easier for doing calculations. If we have a sharp increase, it's very difficult to say, well, at this time point, how many cells are there, because a very small change in this slope means a big change in cells. Over here, we have more lines that intersect and would be easier to calculate. Unlike our test tubes, whether they be broth or slants or our Petri dishes, we being the scientific community, not us at CR, use a chemostat. So stat means constant, chemo is chemical. So maintaining constant chemicals. It's a chamber where we can continually replace the nutrient media. We stir it so that we don't have dead cells or anything else getting all settled at the bottom. And then we have a way to pump out the waste and any dead cells. We can maintain a constant amount of bacteria at a constant level of growth. There are several different ways to determine the number of bacteria present. Now, in our lab, it's typically not really necessary to know how many. We looked at relative turbidity in our growth in broth, but we didn't count the number of cells. In a clinical setting, it might actually be really important to know how many cells are present in a sample, and it's also useful in a research lab, because if we're looking for the production of a particular microbial product or the effect of a drug, it can be really important to know how many bacteria are present. We can directly count them. There's a chamber called the Petrov-Hauser chamber. It has grid lines on it that have a very specific area. The bacteria are in solution that are pipetted onto the slide, and when you look through the microscope, you see this grid. You count the number of cells present, you know how big the grid is, and that tells you the volume. That does count both live and dead bacteria. So we could determine the total number of cells present. If we just needed that biomass, this would be an excellent way to just get how many cells are present. If we needed active metabolizing cells, this would not be a good way, because we would also be counting dead cells. Fluorescence staining can tell the difference between viable and dead bacteria. So the viable cells are green and the dead cells are red. So in this culture, we have a lot more viable cells than dead cells, and we could get, again, a chamber and count up the number of dead versus live, and that would tell us about how many live cells are present. A Coulter counter, because Dr. Coulter figured it out, is a way to use the change in resistance. So if we have the bacteria in this chamber, we can determine the change in resistance, and that will give us a pretty decent estimation of the number of cells. But dead bacteria change the resistance just as much as live ones. So again, it would just tell us the total number of cells, but not the number of viable cells. To count only living cells, there's a process called the viable count. We start with a solution of bacteria. We do a series of dilutions. We start out with sterile media at a certain level. Here it's 9.0 milliliters. We add 1 milliliter and mix it up. That gives us a 1 to 10 dilution. If we take 1 milliliter from a 1 to 10 dilution and dilute it 1 to 10, we get a 1 to 100. And we could keep doing that with only 1, 2, 3, 4 transfers. We get a 1 to 100,000 dilution. This method is used for getting super dilute solutions of bacteria or other chemicals relatively quickly. It'd be hard to take 1 milliliter and put it into 999 99,999 milliliters to get a 1 to 100,000 dilution, but doing 1 milliliter into 9 is reasonable volumes. And if once we do those dilutions, make sure they're well mixed, then we can take a proportion of that and spread it onto an agar surface and count the number of colonies. And if you multiply the number of colonies by the dilution factor, then you can back calculate how many bacteria, how many live bacteria per milliliter were present in the original solution. Again, this would be something that would be useful in a research lab when you're needing to know something about the biomass, something about how many cells survived a particular treatment, and we can just count the colonies that are present and multiply them by the dilution factor to determine that. The ways we can get bacteria onto the surface, we've seen streaking it with a loop, but we can't really count that. We get a huge smear across the surface. But if we have diluted samples, we can mix that diluted sample with some warm molten agar and pour that into the plate and move it around until it's evenly distributed. And then when we incubate that, we can see the colonies and count those up. The other way to get them on a surface is what we would more likely do is have the bacterial dilution and have a little bit of the agar here and spread that onto the surface, and then we get the colonies on the surface. Count the colonies, multiply it by how much we diluted it, and that would tell us how many cells per milliliter of solution we originally had. This is a depiction of the most probable number method. There is a lab late in the semester that I believe we will be able to get to once we complete all the exercises for our unknown. And it is used to determine coliform contamination. Coliforms are gram-negative bacilli, and what got cut off here is that they ferment lactose. So that is an anaerobic process. The sugar is lactose. They ferment lactose and produce both acid and gas. The media contains lactose as the sugar that can be fermented, and it also has a pH indicator so we can see that the acid has been formed. And most importantly, it has little tubes inside the media that is there to trap gas. A similar exercise is our exercise 5-2, the phenol red broth. That is looking at several different sugars. And we're not calculating the relative number of bacteria. We only had one tube of each sugar for each culture. But we could see if acid was made because the media turns from red to yellow. And we can see if gas is made because that interior tube, the Durham tube, collects the carbon dioxide gas that is produced. In the most probable number test, we're looking to do multiple sets with different dilutions of the original sample, and based on the number of positive tubes that have acid and gas produced, we can statistically determine probably how many of the coliform bacteria were present. So we have the media. All of these tubes have the same media. All of them have the tubes for collecting gas. Dilutions of the pond water or the horse trough water or the well water were diluted 1 to 10 and 1 to 100. And then we look at, after 24 hours, how many tubes are positive, how many have gas in them. Because if there's no gas, then even if fermentation happened, it's not a coliform. There must be gas production for it to be a coliform. And this particular pond sample created 5 positive test tubes with the undiluted amount it created 2 positives at a 1 to 10, and it didn't have any positives at a 1 to 100 dilution. So there's definitely coliforms present, and there is a table for doing calculations based on the number of tubes at each dilution that will give you the statistical probability of how many coliform bacteria are present. We can use a spectrophotometer. That is based on light shining through a sample, and how much light gets blocked will be proportionate to how many cells are in there. So clear broth, the light passes entirely through, but turbid broth, only a little bit of light goes through, and a lot of it gets bounced back or absorbed. So proportionately, the more light that gets absorbed, the higher the number of bacteria. However, dead bacteria and live bacteria block light equally well. So this will tell you how many total cells are present, but it will not tell you how many live cells are present. One can take this sample and get the spectrophotometer absorbance reading, how turbid is it, and do the viable plate count, and then you could relate a turbidity measurement to how many live cells does that represent. By itself, the spectrophotometer will tell you total cell population. Do not memorize these. These are short peptides. They are called autoinducers in quorum sensing. To auto-induce means a cell secretes a chemical, and that chemical signals back to the cell that made it. And quorum sensing is a way to determine how many of your own type of cell are in your neighborhood. So that coordinates the response of bacterial cells. So the R side chain of the N-acetylated homoserine lactone is specific for gram-negatives, and they might be recognized by more than one species, but a particular gram-negative cell could secrete this chemical, and it would signal itself. So the more of those species that are in the area secreting this hormone is a way, or this peptide, is a way for the cells to determine or sense how many of them are in a particular area. A pond and some cattle are here to show anaerobic environments, environments lacking oxygen. That doesn't mean there are no oxygen atoms present. Organic matter will always have oxygen atoms in the molecules, but there's no oxygen gas, no O2. So this bog, if it's undisturbed, the mud at the bottom of it will definitely be anaerobic, settled down so that oxygen can't seep into those levels. There are definitely microbes that not only survive but flourish and require that lack of oxygen. And we also have the cattle here to demonstrate the rumen. Cattle have 4 compartments in their stomachs, and so do other ruminants. So the first compartment is an oxygen-free incubator for methanogens and other bacteria that are obligate anaerobes. They require the lack of oxygen. And so those bacteria in the rumen of the cattle's stomach are operating in the absence of oxygen. They are able to help digest cellulose. They produce the enzymes that can digest cellulose. We don't. We don't have a rumen. We don't have enzymes that can digest cellulose. And the methanogens are prokaryotic cells that generate methane. So, cow farts. This slide depicts the growth of various microbes in thioglycolate. This is exercise 2-6 from our lab, growth in thioglycolate broth. That media contains the thioglycolate that binds to any oxygen gas, any O2, and reduces it to water. So reduction is the gain of hydrogens. So if we have starting out with oxygen, we end up with water. We go from O2 to H2O, we gain hydrogens. Obligate aerobes are obligated to have oxygen present and will only grow in the very top where oxygen can diffuse back into that media. Obligate anaerobes will be at the very bottom where no oxygen has been able to diffuse into the media. Facultative anaerobes versus aerotolerant anaerobes. To be facultative means you can do with or without. So facultative anaerobes will survive without oxygen, but we can see that they do not grow as well without oxygen as they do with oxygen. That has to do with the fact that aerobic cellular respiration is way more efficient than anaerobic metabolism. We get 38 ATP for every one single glucose during aerobic cellular respiration. And if these organisms are surviving in the absence of oxygen, but they're fermenting, they're going to get a whopping load of 2 ATP for every glucose. So they will grow much more slowly. Aerotolerant anaerobes grow the same no matter what. And they're called aerotolerant because they grow in the presence of oxygen, but they do not use oxygen. Facultative anaerobes, if you shut off the oxygen, they grow away slowly, but they love the oxygen. They will do aerobic respiration and grow quickly. Aerotolerant ones can only do anaerobic metabolism, and so they're growing where the oxygen is present, but they're not using it, so they're going to grow a lot more slowly than the facultative anaerobes. Lastly, we have the microaerophiles. So phile is a lover of, aero is oxygen, and micro is small. So they are lovers of small amounts of oxygen. So they can't grow without oxygen, but they also cannot grow with high levels of oxygen like in our atmosphere. And so they grow just below the surface. In a face-to-face lab, all of the unknowns are either obligate aerobes or facultative anaerobes. Very hard to maintain the microaerophile conditions, so we don't use that. Also very difficult to maintain obligate anaerobe conditions, so we don't have that. In keeping with that, and for you to be able to use the identification tables that are in Chapter 9 of the LeBoff and Pierce lab manual, we stuck with you're either going to have an obligate aerobe or you're going to have a facultative anaerobe. We saw the videos for using an anaerobe jar. The ones we have are not quite as fancy as this. We don't have a pressure sensor to detect precisely what the carbon dioxide amount is in the chamber, but we did have a pack that would release a chemical to bind the oxygen and create water and create a lack of oxygen environment for our plates. On the right is an anaerobic box. This entire chamber is lacking oxygen. It's not a vacuum. It just does not have any oxygen. So researchers working with obligate anaerobes can keep their cultures within this box so that they are never ever exposed to any oxygen. Hope you weren't eating. This image is shown here. You'll see similar ones when we get to systemic diseases. This is ulcers on the foot of a diabetic patient. These sorts of lesions create an anaerobic environment. The dead skin no longer has oxygen flowing to it, and then the anaerobic bacterium Clostridium perfringens causes gas gangrene. So the gangrene is the dead tissue. There's enormous amounts of swelling in this poor person's foot because gas is being produced as that organism is metabolizing in their skin. Your book will always use the worst possible image for any type of medical condition, so it's pretty obvious which condition it is. But this definitely can happen. And it happens to diabetics because they also are unfortunate to suffer nerve damage. So they may not feel the lesions, they look super painful, but this person probably doesn't have any nervous sensation in their foot. And it can go from not being bad at all to being very, very bad very quickly. The catalase test, detecting the enzyme catalase, which doesn't tell you anything about what the enzyme does, except that it is an enzyme. Catalase catalyzes the breakdown of hydrogen peroxide into water and oxygen. This will be exercise 5-4 in the lab portion. On this slide, there were 2 drops of hydrogen peroxide, and the microbes, 2 different microbes were mixed. A catalase negative microbe with the hydrogen peroxide here, and a catalase positive microbe with the hydrogen peroxide on the other side. A positive result means the catalase broke down the hydrogen peroxide into water, which we wouldn't see because water is clear liquid and so is hydrogen peroxide, but the bubbles indicate that oxygen gas has been made. And the negative one, the organisms are distributed and turbid in the hydrogen peroxide, but there are no bubbles present. Organisms that produce catalase are all either obligate aerobes or facultative anaerobes. It is an enzyme that organisms that are capable of doing aerobic metabolism have to have because hydrogen peroxide can be made during aerobic metabolism, and we need an enzyme that can immediately break that down into water and oxygen. Organisms that do not metabolize in the presence of oxygen don't generate hydrogen peroxide, and so they have no need for an enzyme to break it down. Blood agar looks for hemolysis or blood lysis breaking. The agar contains sheep's blood that is sterilized, and when the organism produces a hemolysin, a protein that breaks down blood, we either see complete clearing around the growth, and that is called beta hemolysis. Where there are no bacteria, the agar remains that deep blood red. On the other side, bacteria have been streaked and pushed or stabbed into the agar, and it's much lighter amount of clearing, and that is called alpha hemolysis or partial hemolysis. We will be doing that late in chapter 5, 5-21, 22, 23, somewhere in there. On the right side, we have a motility test. This one is very difficult to see. This line represents where the bacteria were stabbed into this agar deep, and we don't see anything moving away from it. On the positive one, it's hazy, away from where the original stab was. That indicates motility, that the microbe put into this agar have flagella, and they swam away from where we put it. The motility test we will have with our unknowns will have a dye indicator in it, and the dye will show up only where there is growth, so there would be a straight line of red in this organism, and there'd be swimmy out red in this one. Fermentation that we mentioned in the previous chapter can be used for lots of different food production. We could take the lactose sugar. We undergo this squiggly arrow represents the entire process of glycolysis, and that would make 2-pyruvate. that will happen whether or not oxygen is present. If it's an aerobic metabolizer, it will do glycolysis and produce pyruvate. If it's anaerobic, it will do this process because the enzymes for glycolysis do not depend on the presence of oxygen. However, if this organism finds itself in an environment where there is no oxygen, it cannot continue to the citric acid cycle and electron transport. It will take those pyruvate and do fermentation and make lactic acid. So lactic acid production from fermentation creates things like yogurt, sour cream, cream cheese. We've got pickles, we've got sauerkraut, and we've got, looks like pico de gallo here. Could be fermented or ceviche. That acid curdles the milk. That acid lowers the pH and serves as a preservative. We did or looked at the effect of pH on growth. These are the approximate pH ranges. We can have neutrophiles, neutral lovers, acidophiles that love acid, or alkalophiles that like an alkaline environment. And the best pH is 7, but there is a range of growth for neutrophiles. The best pH is around 3 to 4 for acidophiles, but again, a range. And the best pH is around 10 for an alkalophile. There's always a range of growth, but most living things are going to really appreciate that neutral area, somewhere in that 6 to 8. But it does drop off pretty steeply. A change in pH means a change in the hydrogen ion concentration. The definition of pH is the potential hydrogen ions in solution. If we change the pH, we change the hydrogen ions, and if we change the hydrogen ions, we change hydrogen bonds. Growth depends on enzyme activity. Enzymes are proteins. Proteins depend on hydrogen bonds in their secondary, tertiary, and quaternary folding. So if we disrupt the amount of hydrogen ions present, we disrupt the hydrogen bonding in the enzymes, and that is why the growth is really narrow. Any changes in those hydrogen ion concentrations will affect the active sites of enzymes. This is looking at an alkalophilic. An extreme alkalophilic loves a high pH, and halophilic, salt lovers, that cause this color change in this lake. And these would most likely be in the domain of archaea because of the extreme conditions. This depicts a black smoker at the bottom of the ocean. Super hot, like near boiling temperatures because of the extreme pressure at the bottom of the ocean. There are a lot of very good chemicals down there, and they provide a very extreme environment that most of us would think there's no possible way there'd be life down there. However, there is a lot of varieties of life. We see, looks like some sponge corals around here that are growing just fine, and the microbes that are down there use hydrogen sulfide for energy. They do not require light, so they don't depend on photosynthesizers to have produced any sort of organic matter. They can use hydrogen sulfide gas as an energy source. And that does not fit into our aerobic cellular respiration because there's no oxygen present. It doesn't fit into fermentation. It is a different metabolic pathway called anaerobic respiration. And that is the sum total of what you need to know about that, other than there are organisms that can use an inorganic substance for energy. They will use it for energy to drive the synthesis of organic matter. They have to. They're alive, but they can use inorganic matter as an energy source. The effect of temperature will also change the growth rate. 4 categories here. All of our unknowns are mesophiles. They prefer somewhere from 30 to 37. Psychrophiles, it's important to use that R. They are not psychophiles, they are psychrophiles, are cold lovers, optimally around 10 degrees centigrade. Thermophiles love the heat, looks like around 65. Hyperthermophiles, extreme heat. Their best temperature is close to boiling. That would be those microbes on the previous slide growing in the black smoker. And again, the range is pretty narrow. Most living things do not survive at a very wide range of temperatures, at least not very well. And again, that has to do with the hydrogen bonds of the enzymes. The pH chemically alters those hydrogen bonds by changing the availability of hydrogen ions. Temperature will melt hydrogen bonds. Hydrogen bonds are very delicate. If there's heat, they pull apart. And so heat also affects the hydrogen bonding in the active sites of enzymes, and that is why the growth range is very, very specific to a narrow range of temperatures. This is looking at fertilizer runoff into Lake Erie. A lot of fertilizer. That is going to flood it with excess nitrogen. Excess nitrogen causes algal blooms to form. And that can certainly produce a lot of oxygen, but we know that algae can also produce toxins that can be damaging to the life living in the waters there. And this is the Great Salt Lake in Utah. The purple is from another halobacterium, that it produces this pigment that you can actually see from space. If you ever get a chance, once it's safe to travel to the Great Salt Lake, there is so much salt there that it makes you feel very buoyant in the water. You could be a terrible swimmer and you're going to stay afloat in the Great Salt Lake. If you are a terrible swimmer, wear a life jacket in the Great Salt Lake. But it is really odd to walk into the water and you just let yourself go, and you will float without any effort. The McConkey auger we saw in the tragically confusing exercises 4-1, 4-3, 4-4, and 4-5. Just about got all those images sorted, and we'll send you a message with when those labs will be due. McConkey auger contains lactose as a sugar that can be fermented, and it contains the dye neutral red that will change color if the organism ferments lactose and produces acid. And a positive result, the microbe turns this bright pink. Lactose was fermented and made acid. This organism grew, but it did not ferment, and so it did not produce acid. Gram-positive bacteria we know can grow in various ways, and we had different media that would detect those. This cluster does depend on the environment and the stickiness of their cell walls and the capsules around them to make them grow in these particular clusters. That's it for microbial growth. I did post in the previous module the summary of cellular respiration, breaking it down into the component pathways of glycolysis, the transition step, the citric acid cycle, and electron transport. And I made tables that indicated what goes in and what comes out of each pathway. It's color-coded where the carbon molecules are black. The electron carriers of NAD plus and FAD are in blue, as are NADH and FADH2, the reduced forms of those electron carriers. So all electron carriers are in blue, and the ATP, ADP, and phosphates are all in red. And that matches the color coding in your textbook. The ATP, ADP, and phosphates are the more obvious energy molecules, but the electron carriers, particularly in their reduced state, are also energy molecules. And at the bottom of that document, I put in for the electron transport how we get from that NADH and FADH2 to produce how many ATPs. Those calculations are there. So you can refer to that as the summary for the aerobic cellular respiration.